We're sitting here in the Mabi forest. It was obviously primordial forest, which has been around for hundreds of thousands of years and has ebbed and flowed with ice ages and sea level and so on.
Mabi forest is a rainforest type that is characterized by the presence of deciduous and semi-deciduous trees that occur on basaltic soils. These forests support dozens of endangered and endemic species, such as the Atherton Tablelands. Mabi forests were once the primary ecosystem of the Atherton Tablelands.
Habitation of the tablelands, I think, really started in the century before last, the 19th century, but the land here was really only cleared after about 1930. And this little patch we're sitting in here was not cleared because it was so rocky and it was very hard to clear in those days. They didn't have the big bulldozers, but they did clear all the land around it. And you can see on the aerial photos that there's quite significant clearing and agricultural use. The land was used for farming and market gardens, I think.
Fragmentation like this has negative effects on the ecosystem, other than just decreasing the amount of forest remaining.
Fragmentation actually, it changes a lot the way the animals will movement and the plants as well, in terms of dispersal, and also the microclimate is going to be changed, so which means that we're going to have more light, more wind.
Through clearing, the Mabi has been reduced to just 4% of what it once was. While the Mabi is fairly well studied as a whole, there is a lack of species lists that are specific to certain fragments. These specialized lists are important because each fragment has a unique set of species with a unique set of conservation needs. Knowing the state of a fragment and its needs are important to developing conservation strategies that work best for each fragment. The goal of this project was to create a species list of plants for a Mabi fragment in East Barron, Queensland. This particular fragment is uncleared and had never been surveyed before.
To capture the biodiversity of this fragment, we surveyed the flora of the patch. These include vascular plants like flowers, ferns, and trees. We also paid attention to miniature plants such as mosses and liverworts. These plants are collectively known as bryophytes.
This group of plants is still poorly known and poorly documented in localized checklists. And so if we don't document them, we are not getting the full picture of the natural heritage values of a particular place.
To compile a list of vascular plants, we followed a meander route through the entirety of the 8.6 hectare plot, recording the species encountered and collecting voucher species for a herbarium record. The point-centered quarter method is a survey protocol in which a series of random points are selected throughout the property. At each point, we took record of the diameter, distance, and species of the 4 closest trees.
To describe the full extent of bryophytes in this forest, we utilized floristic habitat sampling, collecting samples from as many substrates and microhabitats as possible, in hopes of capturing as many unique Mabi species as possible. Due to their small size, bryophytes must be identified under a microscope in the lab.
The process for identifying bryophytes is the same kind of process that you would identify any plant or any animal. You need a key, and we have a dichotomous key for bryophytes in Australia, as there are throughout the world. The process, it can sometimes be very arduous and very difficult. There are some genera in the forest here, for example, Physidans. The Physidans moss leaf is easy to identify to genus, but there are over 40 species in the wet tropics. It's a bit tricky.
After over 200 person-hours in the field, we were able to identify 162 unique species of native vascular plants.
Based on the point-centered quarter method, we can describe the forest community as one dominated by a few key species, such as the shiny stinging tree, the red tulip oak, and the Moreton Bay chestnut.
The forest fragment here in East Barron is also home to 42 Queensland endemic species like the Atherton Tablelands, Queensland Videx, and the Atherton turkey bush.
The site also included 4 species listed as endangered or threatened by the Nature Conservation Act of 1992. We collected over 100 samples of bryophytes, and after spending 300 hours at the microscope, we were surprised to find 42 distinct species. Because bryophytes are not a well-studied group, there is not a lot of information on the conservation status of these small plants. We found 7 families of liverworts, with the most diverse being Leguniaceae, and 17 families of moss, with Physidentaceae being the most diverse family. There were only 4 historic records of bryophytes in the Australian Virtual Herbarium from this site. But through our survey, we contributed over 100 more samples from 42 different species.
This project resulted in a complete species list that we were able to give to the landowners of this Mabi fragment, who can use this information to further protect the land under covenant.
It is important for local landowners and land stewards to have access to species lists for remnant forests. To create the most effective land management strategies to conserve Mabi fragments, we must understand specific plant communities.
We hope that this study sets a precedent for well-rounded plant surveys in other Mabi patches around the Atherton Tablelands, so we can continue to conserve and reforest Mabi into the future.